Hello everyone, how are we doing? So, in today's video, we're going to be going into the cell. Well, we're going to be starting first on the outside of the cell where we have the cell membrane. Remember, the cell is selectively permeable and is a phospholipid bilayer. So we're going to talk about the structure of it and how things get in and out of the cell and just how stuff is regulated. And then in the next video, we're going to be going over all the organelles of the cell. And then the last video, we're going to go over transcription, translation, and mitosis. So these next three videos here for the section will be covering all the cellular features that will help us understand anatomy and physiology a little better. All right, so like I said, in this video, we're going to look at the structure and function of the plasma membrane, which is the cell membrane. And then we're going to look into some transport mechanisms like active and passive transport. All righty, so if you recall from the chemistry chapter, from the major macromolecules, we talked about lipids. And one of the classes of lipids, here for lipids are the fats, are phospholipids. Let me spell that correctly, are the phospholipids. So remember, this is a phospholipid, and phospholipids are what make up a cell membrane. So here there are two fatty acids, and here's a double bond showing an unsaturated, so that can vary in each. Here's a saturated, which means it's fully saturated. Then up here is the hydrophilic head, where you have the phosphate group connected to the glycerol head. So this is charged. Charged means it's hydrophilic. Hydrophilic means water loving. Hydrophobic, like a phobia, means water fearing. So water does not want to be near these nonpolar tails. Water wants to be near the polar heads. And the way these line up, some cells are in water. All of the cells in our body are in water. It's an aqueous environment. So for a cell membrane to form, they're made up of these phospholipids. They form this bilayer where the tails are facing inwards and the heads are on the outside and inside. So out here's the extracellular fluid and here's the intracellular fluid, or the ICF. So you have water outside, water inside the cell, and then these tails just turn because they're are repelled in a way from that water where the heads are attracted to the water, so you don't have any water inside. And it forms this phospholipid bilayer. And when we draw a round cell, that is a double layer cell membrane. So any membrane, whenever we say membrane, it's a phospholipid bilayer because that's just the structure of how it is. And there are no bonds that hold these heads together. The only thing that holds cell membranes together are the fact that the heads are hydrophilic and the tails are hydrophobic, and there's the presence of water. If you remove polar water, remember water is polar, if you remove polar water and add a non-polar solvent, the structure would fall apart. So water is one reason, besides many other reasons, that water is essential for life. Now, our cell membranes aren't only made up of these phospholipids. There are also other things embedded in that membrane, like shown in this figure here. The first thing we have are all the different blue clumps here, which are proteins. Some proteins can be just on the outside. They're known as peripheral proteins. So peripheral are on the outside. They could be out on the extracellular fluid out here as well. Some proteins go through and can integrate into the membrane. Like here we have an integral membrane protein. Sometimes these are referred to as transmembrane proteins as well. Some of them can be globular, some of them can be structural, like this one right here. So they can have various structures to them as well. And then you can move things in and out of the cell via these things called channel proteins. So it makes a pore, and so things can move in and things can move out. Not everything can move through the cell membrane. When we talk about active and passive transport down here next, we'll talk about how these things move through via proteins or freely move through. So that's all the proteins. These proteins in the cell membrane act as carriers. You can have a signal for a neurotransmitter bind out here, then send a signal inside the cell. So they can be signal transducers. They can be the channels, enzymes, a whole bunch of different things we'll talk about more throughout the semester. Now, when you put all of these phospholipids together, your cell membranes have a fluid-like structure. And so fluid is very mobile. It's viscous in a way. So your cell membrane has like the viscosity of salad dressing in a way. So to make it more rigid and to sometimes keep proteins together, there's about 10 to 20% of cholesterol embedded in it. Cholesterol adds to the rigidity of that membrane and makes it stronger and more viscous in a way. So cholesterol, very important structural element of the cell membrane. Cholesterol is another lipid, has that four carbon chain with different functional groups coming. Well, cholesterol has a certain functional group coming after it, which we recall from the last video. Alrighty, last little bit we have here are these tails sticking out. These are known as a glycocalyx. So, carbohydrate, glycogen, calyx is tail. So these are sugar or glucose tails. They could be attached directly to the lipids, and they're known as a glycolipid, or they could be attached to a protein, where they're called a glycoprotein. But in general, they're just known as a glycocalyx. So what do these do? These allow your body to hold itself in place, allows your cells to know what's your cells and what's not your cells. So it's called recognizing self, a very, very important part of your immune response. Signaling, adhesion, which I mentioned. The list goes on and on for what these glycoproteins do, but that big major one being immune system. If you get a cell that's foreign from someone else or a microorganism, they will have a different fingerprint on it of these glycocalyxes, and your immune system will recognize that difference and destroy that cell. Or if your cell is struggling on the inside and says, I need to be got destroyed because I'm not functioning properly, it will actually signal via these glycocalyxes to tell the immune system to come destroy it, or it will remove all of them, and that will also signal the immune system to destroy it. I know that's getting really detailed into immunology, which is outside the reach of this course, but just a little hint of the importances of these things sticking out your cell membrane as your body's fingerprint. All right, so there's what makes up the cell membrane. Now, how does stuff move in and out? This is where we go into active and passive transport. The difference is whether or not it requires energy. So first up, we're going to go through passive transport. This is just simple diffusion, facilitated transport, and osmosis, which then leads us into talking about tonicity as well. So passive transport, things are moving from a high concentration to a low concentration. Think about releasing a ball of gas right here. It's just going to fill the room without any energy being put in. But to get that ball of gas back into a high concentration, that doesn't happen naturally, so that requires energy. That would be an example of active. Putting energy in to do something against its natural desire is active transport. Passive is just going to happen passively. No energy input is needed. Things just want to be in equal concentration throughout. So not only concentration here, but also charge concentration. Opposite charges attract. So a positive ion wants to flow towards a negative ion. And if it's in a high positive ion area, it wants to get out of there. We're not going to be getting into the details of that one here today, but it includes both. So what can move through the cell membrane? Small lipids or uncharged molecules and gases can move through. So if you have a high concentration out here, over time, they're just going to move through, and then they're eventually going to be equal inside and out. It's just going to equalize if they can freely move through. But charged ions, small molecules, glucose, things like that, they cannot move through the cell membrane because they are charged. They can't go through the nonpolar area right here. These nonpolar molecules can go through because there's not much interaction. They don't get stuck in here. They just move right through. They don't really interact with the polar heads as much to get repelled by them. There is some repelling, but not as much as the charged molecules. So how do other things move through? Well, they need this is called facilitated transport, meaning a protein facilitates the transport. It could be a channel protein where things like glucose here are moving through. Or they can be a carrier protein where this protein structure here is moving a specific thing in. It could be sodium, chloride, some ion like that, where it's specific for that one thing. And So it binds and then moves through. Whereas up here, it's just a straight channel that's going through. But it still requires that protein, and that channel needs to be open in order for it to work. An example of one of these is called aquaporin. So aquaporin is also a protein channel. So here, aquaporin, it's a protein channel that specifically moves water. So water out here could move in or out of the cell. That's a molecule of water. You would have more phospholipids right here too. And that's just a channel protein. That's no, so aqua is water, porin is pore. It literally means water pore, which is kind of cool. So now thinking of that in more detail, if concentrations want to equalize and we have all of these ions and things like that, glucose dissolved in our fluids, those are known as a solute. When you dissolve something in water, it's a solute. So in a solution, there's the solvent, which is water, and then the solute, which is what's dissolved in there. So you can get a concentration difference of the solutes and the water across the membrane. So this is showing that what if those ions in larger structures could not move? Imagine this was sugar. So you put sugar on this side at a higher concentration, sugar on this side at a lower concentration, and then you put a semi-permeable membrane like the cell membrane in between it. But the pore size was too small for the sugar to go through, but water could go back and forth like an aquaporin. So what happens is you want to decrease the concentration on this side over here. So to do that, water is going to move this way. And as you remove water from this side, it's going to go down and create a higher concentration. Whereas when water is added on this side, it's going to lower this concentration by, you know, adding more water to something. You dilute it, but eventually the concentration on this side will be equal to the concentration on this side, because only water can move. And that's kind of your cells want to be in that state. And this is known as tonicity. Or osmosis specifically is the movement of water, but tonicity is the movement of water in our cell membranes if they're in a different environment. So if everything is equal inside and out, it's known as isotonic. It's an equal movement in and out, and the solute concentration is equal. Now, hyper means above, so we have hypertonic versus hypotonic. So hypertonic is when things move out. So you put the cells in a above, so inside the cell is highly concentrated. So low concentration compared to outside. Outside is a high concentration. So water is going to leave the cell, and then that's going to cause the cell to shrivel. So it's a high concentration of solutes. Where a hypotonic solution below is when you put cells in something like deionized or distilled water. So when that happens, the concentration inside the cells of solutes is higher, so water will move into the cells and cause them to grow. And eventually they could lyse or explode, and rupture. So that's why drinking like deionized distilled water is dangerous. And this is important in anatomy and physiology because this is important for IVs. If you give someone an incorrect tonicity in their IV, you could cause their red blood cells to rupture or shrivel. So either case might not be a good situation for that patient. I remember hypotonic, there's an O in hypo, and these cells could explode, and there's an O in explode. Silly way to remember it, but still important concept. Next up, and the last thing we want to talk about is active transport, which also involves bulk transport, which means we're moving larger and a lot more items at a time. Now, active transport here, the big one means it's using ATP or energy. This is when we're pumping things against their concentration gradient, so they don't want to go that way. The big example we have here is called the sodium potassium pump. You might sometimes see it written as the ATPase. So what happens here, this pump loads in 3 sodium ions from inside. So this is in and this is out. And it pumps them out. They get released. So 3 sodium ions get pumped out of the cell. Then energy is used. 2 potassium ions then load into the cell. This hydrolyzes. That causes the release of energy. And then 2 potassium ions come in to the cell. So this, you might be like, why are we doing this? Why are we pumping 3 sodium ions out and 2 potassium ions in? This is very essential for a lot of processes in our body. Your nervous system does this to make something called resting membrane potential. And it uses that electrical difference, and charge and concentration difference for your nervous system to work. Your muscles use this for potential as well. Your digestive system uses this for absorbing nutrients. Your kidneys use this for regulating sodium and water volume. Very, very important pump right here. We'll get into some of the more details throughout the course, but just introducing you to this pump right now. Just remember 3 sodium out, 2 potassium in. So eventually the concentration of potassium is going to be high inside the cell, and the concentration of sodium is going to be low, and the concentration of sodium is going to be high outside the cell, and the concentration of potassium will be low outside the cell. So you see that over here. And that is an essential part in forming this concentration difference that you can use in secondary active transport in another process. And the last thing we're going to talk about here is bulk transport. There are 3 types of ways you can bulk move things inside the cell. And every time you do that, you form a mini cell or a vesicle because it pinches off part of the cell membrane. The first type here is known as phagocytosis. Phagocytosis is when the cell membrane kind of reaches out, engulfs the large particle, and brings it in. This works for some food particles. It's how your phagocytes and neutrophils and those cells ingest microbes for your immune system and so forth. But the cell actually reaches out these little arms, wraps around it, and then it pinches off and forms this little vacuole. And that confused with things, break it down, and so forth. There are different ways that could go then. Pinocytosis is kind of where the components just end up pushing in, and a piece of the cell membrane just pinches off, and you get that vesicle inside the cell. So this is pinocytosis, or it's known as like cellular gulping, because it almost looks like the cell membrane gets gulped in. And then there's what's called receptor-mediated endocytosis. So whenever you bring something into the cell, it's called endocytosis, entering the cell. If you exit, it's called exocytosis. So receptor-mediated just means there are receptors on the cell, proteins that bind to whatever that's bringing in this case, what's called a ligand. They bind and bring in those specific proteins inside the cell. So they bind, they come in, and they form a coated vesicle that can do certain things inside the cell. Viruses can sometimes get into your cell using these mechanisms. But all of these require lots of energy as well, and they're part of the active transport process. But that's all I have for this video here, going over the cell membrane. Next video, we're going to be going into details on all the organelles. Like always, let me know if you have any questions, but thank you for watching. Bye-bye.